This is going to be partly about my research, what I've been doing in the lab over the last 3 years or so, and partly about what other people have been doing. So an overview of this here in the U.S. and in Europe. So first, just a bit of an introduction to MRSA for those of you who may not live and breathe it like I do. So it does stand for methicillin-resistant Staph aureus. It's a gram-positive bacterium. And this has been on the public health radar for a long time, but mostly as a source of hospital-associated infections. So it's only recently that it's really gotten broader attention. And a lot of us carry Staph aureus. About a third of the population carries some strain, usually in the nose, also occasionally in the throat, skin, and elsewhere on the body. And according to one of the most recent surveys, about 1.5% of the population is colonized with methicillin-resistant Staph aureus. So it's there in the population, although at a fairly low level. And a 2005 report suggests that MRSA is a leading cause of infectious disease mortality. It's estimated to cause about 18,000 deaths per year, again, mostly associated with a hospital, and about 94,000 invasive infections. So those infections are really only the tip of the iceberg as far as MRSA, because that doesn't include a lot of the skin infections that are really the more common manifestation of Staph aureus infections. So why this has gained traction is because it's not a hospital problem only anymore. It has moved into the community. These new strains have emerged that are associated with the community, and that, as I'll talk about today, are associated with farming exposures as well. So this has been in the news a lot. There's been outbreaks in athletic facilities at all levels, from grade school up to professional football. This was just one report in the New England Journal of Medicine about a MRSA outbreak in the locker room or within a professional football team. And they had to do all kinds of things to mitigate that, including, in some cases, complete redesign of their locker rooms to try to eliminate some of the places where they could share MRSA. It's been found in schools, and schools often don't know what to do when they have a student diagnosed with a serious MRSA infection or even a MRSA skin infection where they would be coming to school. So it's really a problem for school nurses that they really don't have a lot of guidance about what to do about this, and it's been such a hot topic in the media that you hear superbug and everyone freaks out. So it's also been found in animals, as will be the topic of discussion today. This is a report from a few years back looking at MRSA, again, from kind of a One Health perspective. A woman had repeated MRSA infections, wasn't able to get rid of them. Her whole family was examined. They were all decolonized if they were found to have MRSA, but they didn't go away. And it wasn't until someone had the brilliant idea of sampling her pets that one of her cats was found to be positive. So the cat was also treated, eventually rid the cat of MRSA, and that stopped the woman's recurrent infections. So, well, the woman's recurrent infections did not recur, so whether that was really due to the cat or not is kind of left open. But animals can have these. Usually in pets, it's transmitted from their owners to the animals. And it can be in other companion animals as well. They found MRSA in horses, as well as food animals. So cattle and pigs will be our topic today. So pig MRSA is fairly new on the scene. This really started with kind of an anomaly in the Netherlands. They found this 6-month-old child that reported to the hospital for a procedure. And the Netherlands has this very strict policy on MRSA, search and destroy. So they swab basically anyone who comes into their hospital, and if they're found to be colonized, they're segregated from the rest of the population to try to keep the levels down. And as such, they have a low level of MRSA in their hospitals and in their population. So it was very strange that this child had MRSA. They didn't have any of the common risk factors like traveling out of the country and being hospitalized previously. So they did an investigation of this. And they found that the child's parents also were colonized with MRSA. And, they couldn't really find any risk factors for them either, but they noted that they lived on a pig farm. And so they started to investigate this. And so they went to a kind of neighborhood meeting where other pig farmers in the area would be present and asked permission and swabbed 26 of them that were there and found that 6 of them were MRSA positive. So this was 23% of that particular population. Remember, here in the U.S., it's only 1.5% of us carry MRSA, and in the Netherlands, it's much, much lower than that. So that was about 760 times higher than what they would expect in the population at large. They also looked at the pigs on this farm. Were only able to culture MRSA from one of them, but then they did some molecular typing and found that the strains in the child, in the parents, in the farmers, and in the pigs all were identical. And my background is I'm a molecular epidemiologist, so I live and breathe also, DNA and all of that. I didn't put a lot of this in here, just kind of as much as is necessary to get the point across. But there's 3 different typing techniques that I'll kind of refer to in here throughout the presentation. One is pulse field gel electrophoresis, and for this, this particular pig strain is non-typable for that. That's where you take an enzyme, chop up the DNA, and run it on a gel so you get this nice molecular fingerprint. Kind of like if you ever watch, CSI or some of the criminal shows, you'll see that, they have all their nice gels up. So that's kind of what that is. So that will give you names like USA 100, USA 300. Those are based on the pulse field gel types. You'll also have a typing technique called multi-locus sequence typing, or MLST. And for that, you take 7 genes throughout the genome, amplify them, sequence them, kind of plug them into a computer algorithm, and it'll spit out your molecular type. And that's where you get things like ST5 and ST398. Then there's a third type called SPA typing, where you look at just one gene, sequence that, and that's kind of a proxy for some of these other more whole genome sequencing techniques. That will give you things like T002, T034. So those are what I'm going to be talking about in here. They're basically all just 3 different ways of getting to the same thing, and what type of bacteria within Staph aureus is that. All right, so on from that. So since this time, swine-associated MRSA have been found in a lot of different countries. Most of these have been ST398, but not all of them, and I'll get into that a little bit later. But when we started our studies in 2007, it hadn't been looked at here in the U.S. at that point. So we're, of course, in Iowa, and people, if they think of Iowa, if they ever do at all, they think it's one of those states kind of in the middle somewhere. But if they know anything about it, usually they know it's for our agriculture, and that is corn and pigs. So those are our 2 big commodities. And we are the #1 pork-producing state in the U.S. About a quarter of the pigs come from Iowa. So our first question was simply, can we find MRSA in Iowa swine? So this started at the end of 2007, looking at 7 farms, mostly in eastern Iowa, but kind of spilling over a little bit into western Illinois. All were part of one production system that had about 60,000 animals roughly at any one time. So we took swabs from a bunch of different age groups, starting at 9 weeks and going all the way up to sows for the adult group. We took 30 per group. We dropped one of them. It got contaminated between the barn and the lab, so we took that one out and ended up culturing 209. And we found that we could isolate MRSA from 147 of those. So that was about 70% of the pigs that we had tested in this system. And you can see that the younger age groups had higher prevalence of MRSA, about 90 to 100%, and starting at 9 weeks and then going up to 15 weeks. And then it seemed to drop off as they aged. So by 24 weeks, only about half of the animals had that. And then the adult sows had the lowest of all. So they had about 36% of them were colonized with MRSA. So again, we did some molecular typing on this. We wanted to see if this was the same thing that they had seen in Europe already. And indeed, we found it was ST398. So that has been kind of the prototypical swine-associated isolate. We also did some antibiotic susceptibility testing. All of these were resistant, of course, to methicillin, makes them MRSA, and penicillin. Also universally resistant to tetracycline for the group we tested. And most were resistant also to clindamycin and erythromycin. Right, so I come into this from the human health side. I'm in the School of Public Health, and it's kind of weird at University of Iowa to be doing like pig testing. That's just not us. That's Iowa State. So we also wanted to look at the people working with them and see if the swine farmers were also carrying this. So at that same farm, we were able to enroll 14 workers, tested them both in the nose and in the throat, and found that 9 of those were positive. So again, we tested a few of these, did further molecular testing, and like the pig samples, these were also ST398. So it suggested there had been transmission events between the pigs and the people on this particular farm. We published this in 2009, along with results from a second farm that was very similar to this first farm, a little bit smaller in size, but pretty much the same antibiotic regimen, and that one was completely negative. We tested pigs and people and didn't find any MRSA on that particular farm. All right, so this did lead to, as Scott mentioned, a pork board-funded study that was looking at multiple states that we did, Iowa and also a little bit of Illinois. Peter worked in Minnesota, and then we had a third colleague at Ohio State, tested in Ohio and also in North Carolina. So the goal of this was to enroll 18 farms per state. We enriched for antibiotic-free farms. We tried to get 50% conventional, 50% antibiotic-free, just so we could, potentially look at differences in antibiotic use between farms. And on each farm, the goal was to test 24 animals, going toward the younger end of the spectrum since we found higher rates of MRSA in those. So we did take nose samples from the pigs, and then again, nose and throat from the humans. In Iowa, we found MRSA in the pigs on 4 out of 9 of the conventional farms that we tested. Didn't find it in any of the antibiotic-free farms. We also found MRSA in 2 additional humans on a 5th farm, but not in the pigs on that particular farm. So total 5 farms where we had MRSA either in the people or the pigs. The overall prevalence in swine for all of those farms was about 10%. But if you look at confinement swine only, it was about 20%. And that goes pretty well with some research in Canada that found swine or MRSA in about 20% of their swine as well. The prevalence in humans overall was about 30%. But again, if you look just at those working in confinement facilities, it was about 44%. So still lower than what we'd seen before, but very high compared to the general population. But in Peter's studies and in Dr. Gabri's studies at Ohio State, they didn't find any MRSA in the pigs. So perhaps this is something that we can discuss a little bit later as far as some thoughts regarding why not. Right, and then again, I'm from the human public health side, and so the thing I want to look at is not only are these people carrying MRSA, is it present, but is it causing any actual infections? Okay. So worldwide, there have been a number of case reports and some mostly hospital-based research looking at ST398 infections in various countries, and they've been found in a number of them, and this is just kind of a brief list of them. You can see it represents mostly countries in Europe, but also some other places as well. So these can be fairly mild, and I'm a microbiologist, so you have to have the gory pictures. Or they can be pretty severe infections. So this was a soft tissue infection in a worker in Belgium. So he was bitten by a pig, waited about 4 days, and eventually went to the emergency room. His forearm was swollen, and it was very tender to the touch. So initially they started him on some cephalosporins and beta-lactams. I can't remember which ones. But then did a culture, found that it was MRSA, switched him over to vancomycin. He had a reaction, so they eventually put him on linezolid for about 2 or 3 weeks. Did some surgery on this as well to clear some of the infected tissue, and eventually he did recover. You can see the second picture. I think that's the 6-month point. So he did recover, has kind of a nasty scar from that though. So they can range from, very kind of mild infections such as abscesses and boils to more severe soft tissue infections to fairly severe invasive infections. And there have been reports of bacteremia and necrotizing fasciitis and ventilator-associated pneumonia. There's also been one report of an ST398 death. And this was actually not MRSA. It was methicillin susceptible, but it was the ST398 strain in France. That was retrospectively found doing kind of a review. So one of the issues with these reports, though, of infections is that many of them are retrospective. So the patient, may have been gone from the hospital for a long time by the time these are found. You don't necessarily have a lot of information about that patient, unless they're obviously, list their occupation as a farmer or something like that, or they're still there, so you can gather that info. Their contact with any live animals is uncertain. So, for some of these, it's thought that perhaps they acquired this in other ways. There has been some recent research that's not published yet that suggests that ST398 actually originated as a human strain, then moved into pigs and probably other animals, and then is coming back into humans. So it may be that there is a low level of ST398 circulating in the human population, and it's these that are causing some of these infections in people that don't have direct contact with livestock. It's also thought that this could be due to food-borne transmission. So people are handling some of these food that may be contaminated with ST398 and picking that up, and that can colonize them and then cause infections at a later date. It could also be due to secondary spread from the community. So one of the things I think Peter will discuss a little bit is, how transmissible is this? If a farmer is carrying this, what are the risks of others in his community that he may transmit it to, his grocer or something like that? So all of these are kind of some thoughts on where people that aren't known to have direct contact with livestock could pick up ST398. I think it's still kind of an open area for investigation as far as which one maybe more important, or if they're all three playing a role. So infections in the U.S., we haven't really seen a lot of these yet. There is some evidence that they're occurring. We sent out a graduate student, actually also in conjunction with the pork board, sent out a survey to pork producers asking them about a number of things, and MRSA infections were one of them. I didn't really expect to see a lot. One of the things we specifically asked about was physician-diagnosed MRSA. So we didn't want them just, lots of people have heard of MRSA. They get a skin infection and they think they have MRSA, but it may not be. So we asked about physician-diagnosed MRSA, and I really didn't think we'd get anything back from that. I come from a farming family. My uncles are all farmers, and they have to be on their deathbed before they'll go and see a doctor, and that's usually with their wife pushing them out the door. So I was kind of surprised that we did have 3.7% of the people that answered the survey say that they had a physician-diagnosed MRSA infection. Of course, the problem with this is that, one, we're taking their word for it, and two, we don't have the samples to type and see if they're, livestock or kind of human strains. We do have another paper with some colleagues in New Jersey that they have a large sample bank of Staph aureus going back about 10 years. And so they combed through that, and they did find 13 ST398 or very closely related infections. So that's under review currently. So they are there, and there's another paper also that I don't have cited here in Missouri at a children's hospital that showed an ST398 infection, although I don't think they kind of realized what that meant because it's not anywhere in the discussion or mentioned anywhere really in the paper besides one figure. All right, so most of this has focused on ST398. And in Europe, that is definitely the dominant strain in pigs. You rarely find anything else other than ST398. But the situation here is different. We found ST398 on our first farm. That was the only type we found of those isolates that we submitted for molecular typing. But in subsequent studies, we found other molecular types. So there's much more diversity here in the U.S., I think, and also some research suggests in Canada, than what we see in Europe. So the problem with this is then trying to define really what is a livestock type of MRSA. Because we've seen, for example, ST5 in pigs, T002 is the spot type, and USA 100 is the pulse field gel type. And this is a common hospital-associated MRSA. So if someone comes in, if a swine farmer would come into the hospital with this type, they would not think to ask him, have you ever had your pigs tested, or is this a risk for you occupationally? They would assume that he probably picked this up in the hospital or something else. Same with USA 300. So USA 300 is the recent community-associated superbug, and we found this in pigs. There was one report of USA 300 in infection in a pig in Wisconsin at the Marshfield Clinic, and then we've also found this in some sampling in pigs in Connecticut, where we found USA 300 spot type T008 and MLST type ST8. What was unique about this is that this was tetracycline resistant. And in humans, you very rarely see tetracycline resistance in human staph isolates. So again, we don't have all the evidence, but that's somewhat suggestive that it may have picked up this resistance on the farm and may be becoming adapted to pigs. And there has been some research in Europe, unpublished, I think, that has experimentally inoculated pigs with ST3 or with USA 300 and found that it lives very happily in pigs, actually even more so than ST398. So that may be an emerging issue as well. So the problem is that, again, when we talk about infections with livestock-associated MRSA, we really have to think beyond ST398 and ways to capture that. And it may be that we need to go to completely whole genomic sequencing, sequence every base pair to really be able to differentiate between human and animal strains. Just one slide on meat, because meat is, of course, one potential way that it can move from farms into the general community. And there have been a number of surveys looking at this. Some of these are listed at the bottom. So it's been found in a lot of meat products. Some samples have been looked at in Canada, a couple studies in the Netherlands, and here in the U.S. Prevalence has typically ranged from about 1 to 12%, although some products seem to be more heavily contaminated than others. In the Netherlands study, for example, turkey had about a 36% prevalence, so it was very high in turkey products. And again, here in the U.S., we see not only this ST398, although that's typically present on meat, but we see a mix of kind of the typical human strains and then the livestock strains. So again, how many of these are due to human contamination during the processing or production process, and how many actually may be coming from the farms. We really don't know that right now. All right, so this is kind of our scheme of what we're looking at. So we have colonized pigs that could potentially transmit that, of course, to other pigs in the farm setting. We know that they can spread this to the farmers, and it's certainly a bidirectional spread, that humans can spread it to pigs as well, and then perhaps it can amplify on the farms. One of the unknowns is really, again, if that can go from farmers into the general community, and if that's an issue we have to deal with or not. And then, of course, the meat products as well, and moving those from the farm to the community. One thing we've looked at also is air sampling. We know that this is present at relatively high levels in the air within these farms as well. So you can't directly just adapt like hospital-associated cleaning procedures and things like that that may deal with MRSA in a hospital setting, but would be completely irrelevant here on the farm settings. And then there have been some issues of, well, can you transmit that to nearby homes or something via the air? I don't think that's an issue, but it's something that really hasn't been investigated very well. And then, of course, airborne transmission can also lead to colonization of farmers. One final thing that we've only looked at in a few samples, but we've also found MRSA in the feed. Coming in, and of course, in the farm, you would expect that if it's in the air, it's, of course, going to be in the feed there. But this was feed coming right off the truck that had never entered the farming facility. So whether that's perhaps a way to introduce this into the farms, I have no idea. Again, it's a small sample, and we haven't followed that up yet. But that was something that we were quite surprised to find. All right, so a lot of unanswered questions, and some of them Peter will address also. So one is the specific role of antibiotics versus other selection pressures. So is this due solely to antibiotic use on the farms, or what other things are playing a role in this? The transmission potential, again, from both directly from animals and also from meat products is pretty much unknown right now. We know that it's in meat. When people have quantitated that, it seems to be at a fairly low level, but we really don't know, how many organisms on that meat sample do you need to colonize someone. And again, here we're not talking about necessarily gastrointestinal illness like with other, E. coli's and salmonellas. We're talking more about food handling, getting on their hands and then, wiping their nose or something like that and inoculating themselves with Staph aureus. And then the ecology of this, and that's one big area that I really would like to go into at some point here if someone will throw me some money. So this was one plate we did for some very low-tech air sampling. We basically put this plate, this is a selective plate for MRSA and other methicillin-resistant organisms, left it in the barn with the lid off for 5 minutes just to see what would settle down on there and what we could pull out of the air. So I have a very old, bad camera, so you'll have to excuse the photography. But these here are the pink colonies that are circled, and you can really not see very many of them. But there are 4 of those on the plate. Okay, so we tested those. Those were all MRSA. Okay, on this particular plate, they turn kind of a mauve color. So for MRSA, out of all of these colonies on this plate, okay, so MRSA is very obviously, kind of the needle in the haystack, and there's a lot more going on as far as organisms there that contain the methicillin-resistant gene cassette. And most of these, we only tested about 5 or 10. Most of them were other species of coagulase-negative Staph aureus, or coagulase-negative staph. So again, those are acting probably as a reservoir, probably are on some of the farms also where, maybe MRSA hasn't been detected yet, but maybe in the future they would lend their genes to Staph aureus and you could have development of MRSA there. So I think you need to better understand what else is there besides just Staph aureus. All right, so to sum up, we do know that MRSA is present in U.S. swine and swine workers. How that is distributed across the country, we really don't know yet. Whether there is some geographic variation in prevalence. We know that there are a lot of types besides ST398. So when you think of livestock-associated MRSA, don't only think of that one particular type. And there is the potential for transmission via a lot of different routes, but whether any of them actually occur, and if so, how frequently, is still kind of an open question. Okay. And so those are my lab numbers.